Most diets treat fat loss as one single problem. Eat less, move more, burn more than you consume. And that logic works to a point. But here is the part almost nobody talks about. A calorie deficit burns fat from everywhere in your body. Your face slims down first. Your arms follow. Your legs shrink. And visceral fat, the fat wrapped tightly around your liver, pancreas, and intestines, is often the very last to move. Even after months of doing everything right. This is not a failure of effort. It is a biology problem. Visceral fat is not ordinary fat. It is metabolically active tissue that functions almost like a rogue organ inside your body. It secretes inflammatory chemicals continuously. It worsens insulin resistance in the surrounding tissue. It produces its own hormonal signals that tell your body to protect and preserve it. And it has its own cortisol receptors, roughly 3 times more concentrated than in fat tissue elsewhere, making it uniquely sensitive to stress hormone signals that promote storage. This is why standard dieting leaves visceral fat stubbornly behind, even when the rest of your body has visibly changed. To directly target visceral fat, you need more than a calorie deficit. You need foods that intervene in the specific biological mechanisms keeping it in place, not through cutting calories alone, but through direct biochemical action on the tissue itself. These 5 foods have research behind them, not general weight loss research. Research specifically showing measurable reductions in visceral fat through distinct biological pathways. Food number 1, fatty fish, salmon, mackerel, sardines, trout, and herring contain omega-3 fatty acids, specifically EPA and DHA. Your body cannot produce these on its own, and these 2 molecules do something that almost no other dietary fat can do. They compete directly with cortisol at the receptor sites on visceral fat cells. Cortisol is one of the primary hormones that tells visceral fat to hold its position. When EPA and DHA occupy those receptor sites instead, the cortisol signal weakens. The instruction to store and protect visceral fat becomes quieter. Omega-3s also activate a protein called PPAR-alpha inside fat cells. PPAR-alpha acts almost like a switch that tells visceral fat cells to shift from storage mode into oxidation mode, into burning mode. This is a direct molecular intervention on the tissue itself, not just a side effect of eating fewer calories. A comprehensive review published in Obesity Reviews confirmed this. Omega-3 supplementation, combined with exercise, reduced visceral fat significantly more than exercise alone. The omega-3s were not simply cutting calories. They were changing how visceral fat responded to the hormonal environment around it. Aim for 2 to 3 servings of fatty fish every week. Canned sardines and canned salmon offer nearly identical nutritional value at a fraction of the cost of fresh fish. The research does not distinguish between fresh and canned. What matters is the omega-3 content, and that is preserved in both. Food number 2, avocado. Avocados work on visceral fat through 2 separate mechanisms simultaneously, and both are worth understanding. The first mechanism runs through your gut microbiome. Avocados are one of the richest sources of soluble fiber available, with roughly 10 grams per fruit. That soluble fiber feeds beneficial gut bacteria, particularly strains that produce a short-chain fatty acid called butyrate. Butyrate has a direct and measurable effect on insulin sensitivity at the cellular level. When your cells respond to insulin more efficiently, your body needs to produce less of it after each meal. And lower insulin means a weaker fat storage signal being sent to visceral fat throughout the day. The second mechanism runs through satiety hormones. The monounsaturated fat in avocados stimulates the release of cholecystokinin, a hormone that signals fullness to your brain. When cholecystokinin rises after eating avocado, your appetite stabilizes, you eat less at subsequent meals, and your insulin response over the course of the day stays flatter. A study published in the Journal of Nutrition followed over 100 overweight adults for 12 weeks. The group eating avocado daily showed significantly greater reductions in visceral fat compared to the control group. Importantly, both groups consumed the same total calories. The avocado group was not eating less overall. They were eating differently, and their visceral fat responded to the difference. Half an avocado added to your first meal of the day captures most of these benefits consistently. sliced onto eggs, added to a protein bowl, or simply eaten alongside whatever you normally have for breakfast. Food number 3, cruciferous vegetables. Broccoli, Brussels sprouts, cauliflower, and kale contain compounds that most people have never heard of. But those compounds are doing something specific inside your body that directly affects where fat is stored. The first compound is indole-3-carbinol. When your digestive system processes this molecule, it converts it into something called DIM. DIM is a natural estrogen traffic director. It helps route excess estrogen toward a weaker, easily excreted form, rather than allowing potent estrogen to recirculate. This matters for visceral fat for a reason most people are never told. Visceral fat tissue produces its own estrogen. The more visceral fat you carry, the more estrogen is generated locally. The more estrogen accumulates, the stronger the signal to store more fat in the same location. It is a self-reinforcing cycle that calorie restriction alone rarely breaks. DIM from cruciferous vegetables helps interrupt this cycle by accelerating estrogen clearance before it can recirculate and strengthen the storage signal. The second compound is sulforaphane. Sulforaphane activates a cellular defense system called NRF2. NRF2 is one of the most powerful anti-inflammatory switches available to human cells. When it is activated, cells become significantly better at managing oxidative stress and suppressing the chronic, low-grade inflammation that keeps visceral fat metabolically entrenched. Visceral fat produces inflammatory cytokines continuously. Those cytokines make the surrounding tissue more insulin-resistant, which in turn drives more fat storage. Sulforaphane does not just reduce general inflammation. It specifically targets the inflammatory environment that visceral fat creates around itself. A clinical trial examining regular sulforaphane consumption found measurable reductions in inflammatory markers specifically associated with visceral fat after 12 weeks of consistent intake. Aim for 3 to 4 servings of cruciferous vegetables per week. One important preparation note. Overcooking significantly reduces sulforaphane content. Light steaming until just tender or roasting at moderate heat preserves far more of the beneficial compounds than boiling until soft. There is also a simple trick worth knowing for maximizing sulforaphane. After you chop or crush cruciferous vegetables, let them sit for 5 to 10 minutes before cooking. This waiting period allows an enzyme called myrosinase to convert more glucoraphanin into active sulforaphane before heat deactivates the enzyme. A small preparation habit that meaningfully increases the anti-inflammatory benefit of the same amount of food. Food number 4, green tea. Green tea contains a compound called EGCG, epigallocatechin galate. EGCG does 2 things simultaneously that directly affect visceral fat, and both mechanisms are distinct from simply reducing calorie intake. The first mechanism works through an enzyme called AMPK. AMPK is sometimes called the metabolic master switch of the cell. When AMPK is activated, cells shift their priority from storing energy to using it. They increase fat oxidation, decrease fat synthesis, and improve their responsiveness to insulin. Visceral fat is highly metabolically active tissue. It responds to changes in cellular signaling more readily than other fat depots. When EGCG activates AMPK, visceral fat cells receive a direct instruction to burn rather than store. The second mechanism works through an enzyme called catechol O-methyltransferase. This enzyme normally breaks down norepinephrine, a hormone that signals fat cells to release their stored energy. When EGCG inhibits this enzyme, norepinephrine stays active longer. Fat cells receive the release signal for an extended period, instead of having it shut down quickly. The research on this is unusually specific and convincing. A 12-week study using CT imaging scans tracked participants drinking the equivalent of 4 to 5 cups of green tea daily. Visceral fat decreased measurably in this group. Total body weight stayed essentially unchanged. That distinction is critical. The fat was disappearing specifically from around the organs, not from general body weight reduction. This is direct targeting, not calorie restriction. 3 to 5 cups daily captures the research-supported dose. Drink it unsweetened and brew it fresh, rather than relying on bottled versions which typically contain added sugar that counteracts the metabolic benefit. Drinking it 30 minutes before a meal adds an additional benefit of blunting the insulin response to that meal. Food number 5, apple cider vinegar. Apple cider vinegar is probably the most misunderstood food on this list. It is either dismissed as a wellness trend or overclaimed as a miracle cure. The reality sits somewhere more interesting than either of those positions. The active compound is acetic acid. Acetic acid works through a mechanism that directly affects how your body handles energy at the cellular level. Like EGCG in green tea, acetic acid activates AMPK. When AMPK is triggered by acetic acid, the same cascade follows. Cells shift toward burning stored energy rather than adding to it. Visceral fat, being highly responsive to cellular signaling, receives a more direct impact from this shift than subcutaneous fat does. But acetic acid has a second mechanism that operates through a completely different pathway. It slows the rate at which food leaves your stomach. When gastric emptying slows, glucose enters your bloodstream more gradually after a meal. Insulin does not need to spike as high to manage the same glucose load. And as that blunted insulin spike is repeated across every meal every day, the cumulative effect on the insulin environment is significant. Lower average daily insulin means fewer hours in fat storage mode. Fewer hours in fat storage mode means more hours when visceral fat can be released and used for energy. A study published in the Journal of Functional Foods found that consuming vinegar before a high-carbohydrate meal reduced the post-meal blood sugar spike by up to 20%. The insulin response dropped by up to 19% in the same subjects. A separate study from Arizona State University found that consuming apple cider vinegar before bed lowered fasting blood sugar by 4 to 6% the following morning. These are not general weight loss effects. They are specific metabolic changes that directly alter the hormonal environment that visceral fat depends on for its persistence. 1 tablespoon diluted in a full glass of water, taken 10 minutes before your largest meal, captures the documented effect. If your stomach is sensitive, start with half a tablespoon and increase gradually. So let's bring all 5 of these together. Fatty fish provide omega-3 fatty acids that compete with cortisol at visceral fat receptor sites and activate the fat oxidation switch inside visceral fat cells directly. Avocado provides soluble fiber that improves insulin sensitivity through gut microbiome health and stimulates fullness hormones that flatten the daily insulin curve. Cruciferous vegetables provide indole 3-carbinol that supports estrogen clearance and sulforaphane that suppresses the inflammatory environment visceral fat creates around itself. Green tea provides EGCG that activates AMPK to shift cells toward burning and inhibits the enzyme that shuts down fat release signals prematurely. And apple cider vinegar provides acetic acid that activates AMPK through a second independent pathway, while simultaneously slowing gastric emptying to blunt insulin spikes at every meal. 5 foods, 5 distinct biological mechanisms, all converging on the same target. The visceral fat that calorie restriction alone consistently leaves behind. None of these need to be added all at once. Pick the 2 or 3 that fit most naturally into how you already eat. Work them in consistently for 30 days. Then add the others if you want to go further. Consistency with a few of these will outperform perfect compliance with all 5 for 1 week, followed by abandonment. There is also a compounding dimension worth understanding here. These 5 foods do not just work in parallel, several of them work synergistically. Fatty fish lowers cortisol signaling at visceral fat receptors, while green tea activates AMPK to increase fat release. Both mechanisms running simultaneously create a stronger combined effect than either alone. Avocado improves insulin sensitivity through the gut, while apple cider vinegar blunts insulin spikes at every meal. 2 different pathways, both converging on lower daily insulin, which is the single most important hormonal condition for visceral fat release. And cruciferous vegetables reduce the inflammatory environment that makes visceral fat resistant to all other interventions. Addressing inflammation is the prerequisite that makes everything else work better. Think of these 5 not as 5 separate solutions, but as 5 levers that all pull in the same direction. The more of them you apply consistently, the stronger the combined signal your body receives to finally release what it has been holding on to. If this gave you a clearer picture of how specific foods can directly target visceral fat, rather than just reducing general body weight, go ahead and hit the like button. And tell me in the comments, which of these 5 foods are you already eating consistently? And which one are you adding first? Because choosing the right foods is not just about eating less. It is about giving your body the specific signals it needs to finally release the fat it has been holding on to.